Hi students, welcome back to my channel NEET Target 2023. So in this video we are going to see few important questions in the lesson Biotechnology and its application. So the first question, how can we say that the copy number of the plasmid vector and the yield of the recombinant protein are related to each other? So the answer for this, the copy number of the plasmid vector is directly related to the yield of the recombinant protein. Higher the copy number of the vector plasmid, greater is the copy number of the gene, and consequently the yield of the recombinant protein is in higher amounts. So this is the answer. Second question, can the exonuclease enzyme be used while producing the recombinant DNA molecule? No, because the exonuclease cannot be used while producing the recombinant DNA. This is due to the exonuclease degrading the DNA molecules. It cannot produce the DNA fragments with the sticky ends. Third question, write the features of the plasmid being used as the cloning vector. So what are the features of the plasmid used as the cloning vector? They are, the cloning vector should have an origin of replication. The cloning vector should have a selectable marker. The cloning vector should have restriction sites. So these are the characteristics. Fourth question, define competent cells. What is the meaning of the word competent? So the answer is, the competent cells are those which allow the foreign DNA to incorporate into the host by the slight alteration in the cell wall. The competent means the ability of a cell to intake the foreign DNA. This is the meaning for the competent word. So it should do some alteration in the cell wall and also will allow the foreign DNA to incorporate into the host by this alteration. So the competent means the ability of a cell to intake the foreign DNA. Okay, now fifth question. What do you mean by eco, R and I to in the enzyme ecori? So the word eco means the species from which it is taken, R means to the particular strain, and I means that it was the first enzyme isolated from the strain. So this is what the meaning of eco, R and I in the enzyme ecori. Next, why are the proteases enzyme added when we isolate the DNA? So why the protease enzyme are added while we are isolating the DNA? The proteases are added to degrade the proteins. So from the name of the enzyme itself you can understand proteases will degrade the proteins so that they do not interfere with the downstream of the DNA treatment. So thus the protease enzymes are added. Seventh question, when the denaturation step is missing during the PCR, what will be its effect on the entire process? So when the denaturation process is missed, there will be no separation of the DNA strands. The primers will not anneal to the template strands, and eventually the amplification of the DNA will not occur. Next question, eighth question, write the name of a recombinant vaccine. The hepatitis B vaccine is a recombinant vaccine. And students, a quiz is available on this topic. Who all wish to attend the quiz can register yourself, join yourself in the Telegram group. I have given the Telegram group link in the description. Next question, ninth one, how can we say that the TI plasmid of Agrobacterium tumefaciens modified to convert it into a cloning vector? So the TI plasmid is a tumor inducing plasmid right? That you know. The genes responsible for its pathogenic nature are either they removed or altered so that it does not they harm the plants and the only delivers the gene of interest. So this is the thing. Next, ninth question. How can we say that the TA plasmids of Agrobacterium tuberculosis modified to convert into a cloning media? Just now we saw this question. So 10th question. Do biomolecules such as the DNA proteins exhibit the biological activity in the anhydrous condition? No, the biomolecules do not exhibit the biological activity in anhydrous condition. That is why the life does not sustain without the water. 11th question. Define biotechnology. Biotechnology is defined as the broad area of the biology that uses both the technology and the application of the living organisms and their components to develop, modify, and to produce the useful product for the human welfare. The term biotechnology was coined in the year 1990 by an agricultural engineer, Karoli Erki. Therefore, he is known as the father of biotechnology. So, he is the father of biotechnology also. 12th question. Define genetic engineering. The genetic engineering is the technique mainly used to change or to modify genetic material, DNA or RNA, and to introduce them into the other organisms. Next question. Write the principles of the biotechnology. The modern biotechnology started with the two crucial technologies, that is genetic engineering and chemical engineering. 14th question. How can we say that the biotechnology is useful in the developing food crops and in the agriculture progress? The biotechnology extends its application over the broad spectrum in the field of agriculture and the development of food crops. In the agriculture field, it helps in the improving food quality, quantity, and the processing. The biofertilizers and biopesticides are the eco-friendly sources for agriculture that contains the living microorganisms, which helps in promoting the growth by increasing the supply or the availability of primary nutrients. The farmers choose biotech crops to increase the yield and in the lower production. So far we have seen 14 questions, which is very important in this topic. So revise it again and again. Go through the video once again. Like, comment, share and subscribe my channel. Thank you.